So I asked you, how are we going to shorten our goats essentially over a 20-year process? Let's see what you guys told me. This is where it gets silly. Oh, we're having fundraiser to buy. Oh, there you go. Yeah. A little crowdsourcing. Only allow shorter-legged goats to reproduce. Yeah, like just eat the tall ones. Right? Okay. Yeah, which is what we're talking about, artificial selection in a way, right? Send them to the wild. Let the wild ones roam. Okay. That's a book, isn't it? Where the wild things roam. Yeah. Use the barn as a fencing and take down the barn and cut the wood pieces down. Oh, there you go. Recycle reuse. Reuse the barn. Repurpose it. Just breed the short goats. Cut their legs off, but now we're getting dark. All right. Use artificial selection. Yeah. That was, I don't know. I don't think tanning's going to help them be any shorter. No, see how we just got the rolling pin and went right over that and like flattened right out? No. Chop off the legs. Oh my gosh, what in the world? Oh. Stuff them? Stuffing things? Oh my word. Reduce their food and starve them. Okay. A lot of killing going on here. Okay. Yeah, right? You know what I'd do? Just get some rope and hobble them. I mean, you don't have to, like, kill them. You know what I mean? Just tie their legs together. They can't jump then, right? It's like a goat concentration camp. Just goats hobbling around, can't run. Yeah. Plus, they wouldn't be able to work out as much. That meat would be more well marbled when you did eat them. Yeah. No? Yeah. Now we're getting dark again. Okay. So, that's fun. Now I have like miniature goat Auschwitz in my head. So we're going to talk about today microevolution and how we study it using a bell curve. Today's really short. We'll talk about phenotypic variation, how we measure that. And that leads to genetic variation. And finally, how genetic variation originates. We know the source of all new alleles is a mutation. But we're going to look at frequency and allele frequency, Todd, today. So Charles Darwin knew that heredity influences characteristics, but he did not know about genes or alleles, right? He was missing that genetic component. I think Levi Strauss came around the early 1900s. Right. When Gene showed up. So we can now study and predict genetic variation. Actually, I don't know when it came out. It might have been before that. I got to look at the back of my jeans when I get home. Every time I wonder that, I'm wearing the jeans and I can't see, And you read, the date's all backwards in the mirror. And you can't see. Then people look at you funny when they walk in the bathroom and you're like looking at your butt in the mirror. It's awkward for everybody at that point. Yeah. So we can now study and predict genetic variation and change the underlying evolution. We do this using genetic frequency, allele frequency. So microevolution is evolution at the level of genetic change in populations. So now we're talking about how do we measure microevolution? What counting method do we use? We're going to do a simulation with the peppered moth. Anybody ever heard of the peppered moth? No? Okay. Industrial melanism. Sometimes in middle school, you might not have done this yet. That's okay. So microevolution can be studied by observing changes in the numbers and types of alleles in a population called population genetics. So if I'm studying the frequency and the amount of different versions of a gene called alleles in a population, like how many brown-haired people there are, how many blondes there are, this is what's called population genetics. And the study of both genetics and evolution kind of go hand in hand, so they advance together. And in the link from micro to macroevolution with speciation, now we can study this in detail and see when do you genetically become different than another one of, let's say, your cousins or your past ancestral cousins. We have our own evolution of modern man. This is like going from Australopithecus to Homo erectus to modern man. And so we understand we have all these, we think of them as cavemen, right? But there's even earlier versions called hominids of us too, even before we get to like what we think of as being a chimpanzee or another primate. These are early versions of man. Now the variety of phenotypes that exist for a given characteristic depend on how many genes affect it. So we understand that our height is influenced by thousands of genes, but polygenic characters are going to be influenced by several genes like eye color and height. So most human characteristics, we cannot just say they are monogenic, caused by one gene. Pea plants are different, right? Pea plants, we can look at characteristics that were influenced by only one gene. But biologists, we study polygenic phenotypes by measuring each individual in the population and then analyzing the distribution of the measurement. So how often does, let's say, the blonde-haired gene show up in a population of 1 million people? And then you can actually give it a numerical value at that point and determine allele frequency. Can you always determine genotype from phenotype? No, but you can sometimes, right? Like if it's homozygous recessive. So a distribution is a basic overview of the relative frequency and range of a set of values. Oh, and you think range, and you guys automatically go, oh, I know what that is. That's my mean, median, and mode, right? Range data distribution. You guys remember that from math class? Yeah. The bell curve, the mean, median, range. Mean, median, mode of a range of data distribution? No? So often some values in a range are more common than others, but a normal distribution is what's known as a bell curve. And this tends to cluster around an average or a mean in the center of the range. I always remember it's mean to call somebody average, right? I remember average and mean go together. You guys remember what the mode is? The most common one? Yeah? Yeah? No. Oh, no. I won't do that to you with math and science together. It's just mean. That's just average. It's like my new, that'll replace basic. We'll just call people, mean. But it'll have like an extra meaning to it. Right? Sorry. That's just going really bad now. Y? Y-axis right here. Okay. It's the end of the year. Just go all in. All right, you guys. A normal distribution will look like this. In this case, we're looking at the height distribution, and we're looking at it in a measurement of meters. So in this case, this must be the pygmy population, because the average is like 1.45, somewhere in there. Okay. And you see there are very few very short and very few very tall, but you see in the middle you have your average height being about 1.5 meters. Now, a histogram is more of a bar graph type piece of data, whereas a actual curve is going to be a line put on that across that histogram with points in it. So I'll probably have you guys do a regular line graph when we do our bell curves in lab. Could this bell curve shift one way or the other? What if all of a sudden we had a drought and the fruit was higher up in the tree? What do you think would happen to this bell curve? Would it go right or left? Let me ask you that. Will this curve shift left or right? Well, would height increase or decrease? Right for increase, left for decrease, if the fruit on the tree were higher this year, the next year? What would happen to this? If you go up, it's right, down, it's left. Just as you're looking at it. I know I've confused you all now. So it would be up or right, meaning the people got taller, right? Okay. Exactly. So what would happen is this, and we'll talk more about this the next lecture. But what happened there? But if, let's say, one year the food source was higher up in a tree, this bell curve would shift directionally to the right. Now this makes me think of our finches we talked about, right, where their beak size shifted from year to year. Now imagine if the trees just kept staying in one direction or one height, then people would generally be taller, right? And so think about now if you only have one type of food source and our shift heads in one direction and not the other direction eventually will go away. Does that make sense? And then all of a sudden all you'll be left with is taller, in this case, individuals. And you'll no longer have the short version. So I mentioned early man and hominids. The early hominids are extremely short. Are there extremely short hominids anymore? No. So that we've gone so far away from that as far as a bell curve in our average human that we're no longer early hominids. And the same thing can say about our skull size. Our skull brain and our brain has changed, and our skull has actually changed as well. And if you look at even early man's skull and our skull now, our brain has grown and shrunk in different places based on how we use it, and we've shifted away as far as the size or the shape of our skull is too. And so we can look at this in a lot of different ways in a lot of different populations. But basically we're talking about changing our average. And we will discuss that a lot more when we get into the different types of bell curve shifts that we can see. So the particular combination of alleles in a population at any one point in time makes up what's called the gene pool. In this room, we would have a gene pool. So if we should go swimming. That sounds like fun. Throw you all in a pool, then we'd have a true gene pool. Only if we were all wearing denim, though. So if we look around this room and I were to check out the different alleles that you all have, and we kind of could write them down and count how many like blonde hair and how many brown hair, how many blue eye, how many, that would be our gene pool. We can refer to a gene pool as a room, as a state, as a country, as a continent, as a world. We can do it in a lot of different ways and at a lot of different levels. But we measure it in what's called frequency of alleles. How often does an allele show up, or a version of a gene show up within that gene pool? So a frequency is the proportionate ratio of a group that is of one type. To study genetic change, the frequency of each allele in a population can be tracked over time. Raise your hand if you have blue eyes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So like 12 out of like 25 people have blue eyes in here. That means I have 24 blue-eyed alleles out of my 50 possible alleles, right? Which is really weird for a recessive trait, isn't it? Think about it. It's supposed to be less common, isn't it? What's the name of the city we live in? Bay City? This is like a Polish-German community, right? Ah, makes sense now, right? If you go to other places on the planet, this is not the way it is. This is unique to Bay City, by the way. Even go to other places in Michigan, that's not the norm. You never thought of that, did you? Take a look around, and then like go to. I'll tell you I'm not from here. I'm from northern Michigan, Traverse City, way up here. There are less blue-eyed and blonde-haired people up there for sure. It's weird. Not weird. It's just you don't think of it until you're like immersed in it. When I first came here, I couldn't pronounce anybody's name either. That was terrible. I was like, oh my gosh, what is this Insky thing? I don't know how to say it. I would just murder. I still do. I murder kids' names left and right. I don't know how to do it. What? That where you are. Oh. Well, I noticed it because I'm not from here. It was like, whoa, there's a lot of like blonde-haired, blue-eyed people running around here. And you don't really think of it until you start thinking about the genetics behind it, right? Now, blonde hair is not recessive, but blue eyes for sure is. All right. So we can actually track alleles, right? You guys all just raised your hand. I counted how many homozygous recessive alleles we had in the room, and that's not even counting the heterozygous individuals. I'm heterozygous, okay? So I carry a blue eye trait too. Hey guys, What are the chances of me having a blue-eye trait? Everybody say 50-50. 50-50. Now, without doing my pedigree, obviously, but if I know my homozygous recessive alleles and I know it's 50-50 that I could be heterozygous or homozygous dominant, can I statistically analyze my population's alleles at that point? Yes. So this is how we study frequency, Mr. Todd. We use a little bit of algebra. Pretty simple. So what does that mean? That means if I count around the room, and I'll even add our data to this chart, okay? So we counted the room, and we said, I think I counted 12, which is 24, right? Why do I say 24? Anybody know? Because there's two, right? One from mom, one from dad. And then everybody else we're going to say is either homozygous dominant or homozygous recessive. Now, I know it's not that simple, but let's pretend it is, okay? And so that means 24 out of our possible 50. Let's make it real easy. Let's say that's 50%. Okay, let's round because it's pretty close. Okay, so that's 50%. That means I would have 25% here and 25% here, right? Did you guys get that? Okay. And if I were to proportionalize that, I would just invert that back and say, okay, well, that means that if I have 24 that are here, that means that I would have like 12 and 12 as far as my numbers of dominant and recessive. So that would have to be 12 and 12, and that would have to be 12 and 12, right? Is this making sense to you? Okay. If I add everything together, it will always equal 1 or 100%, right? And that's why. Basic algebra. Looking at a percentage as a whole being 1, then breaking it down from there. Based on what you do know, not on what you don't know. Everybody say Hardy-Weinberg equation. Hardy-Weinberg equation. That's all. That's what that is. We do it a lot more in AP Bio. We don't have to worry about it as much in regular bio, but in AP Bio, I would give you like a genetics problem. You would have to determine allele frequency, put it into a chi-square, and determine your degrees of freedom, how accurate you were. So. Now, evolution cannot proceed if there is no variation. The major source of new alleles in natural populations is mutation in germ cells. Why do I only look at mutations? Oh, my clicker's not going to work today. Why do I only look at mutations in my germ cells? Why would I only look at mutations in germ cells? Because they're gross, they're germs, right? No, that's not. Why would I only look at mutations in germ cells? 5, 4, 3, oh no. Lock you out like the Taliban. There we go. Only they get mutated first to exist, kind of. They reproduce better, close. Only mutations in germ cells are passed on. Germ cells, let's just talk about germ cells. Because they are the reproductive cells, yes. They're the one, well, not, any cell can mutate. So, I can tell we're a little off base here, majoritarily speaking. Germ cells are found in gonads, testicles, and ovaries. Ms. Lorenz, are we focused? Okay. So, in order for something to be passed on, it must be in a sperm or an egg, right? So germ cells become sperm and egg through meiotic divisions, meiosis 1 and meiosis 2. If I mutate my skin, it is not going to be passed on to my children, okay? We do not reproduce with our skin cells. We reproduce with germ cells that divide and become sperm and egg. So just any mutation in your body is not going to be passed on. That's why cancer generally is not inheritable. Okay? You can't pass that on because usually cancer is of body or somatic cells. So mutation generates new alleles at a slow rate, and it has to be a mutation in a germ cell, bless you. And only mutations in those germ cells, egg and sperm, are passed on to the offspring. So you could have millions of mutations, but you could have a mutation that would make your child a super baby, but unless it was in your germ cell, it's not going to be passed on. And then it would have to be the germ cell, one of the 4 million sperm that get made. It'd have to be the right one that gets passed on. So now you start to really think about how slow this evolutionary process must really be. So microevolution is studied by observing changes in the numbers and types of alleles in a population, counting them in terms of frequency, how often they show up. Biologists study polygenic phenotypes by measuring each individual in the population and then analyzing the distribution of measurements in a range, a distributional range, called a bell curve. And genetic variation and change are measured in terms of the frequency of alleles in a gene pool of the population. Remember, Hardy-Weinberg states that all alleles will add up to 1. And then if you want to figure things out, just look at the homozygous recessive individuals, and that will give you a good starting point to figure out the rest. And the major source of new alleles in natural populations is mutation in germ cells, not somatic cells. Now, there is a review that will be on Skyward. Tomorrow they have me next door at the ISD for Technology Fun Day. Okay? Not really. So, I'm not going to assign your keys today, but if you want to get ahead, go for it. I'm going to leave your vocabulary here tomorrow for you to work on. So tomorrow you're to work on vocab, keys, and finish this review if you have to. All right? Sound like a good plan? Nothing crazy, nothing new. We're done with our CCR. We don't have to. You want one more? Okay. All right. I'll leave one. I'll make one up today. Don't worry. It'll be here.